My name is Josh Millstein, and I'm a faculty member in the Department of Population and Public Health Sciences at the Keck School of Medicine of USC. My research focus involves developing and applying statistical methods designed for studies that generate large-scale data sets that include a genomic component. So an objective of the study might be to look at DNA variation across all chromosomes and identify particular variations that are associated with risk of disease, disease progression, or response to treatment. And the hope is that this new knowledge would increase our knowledge about disease and the disease process and eventually lead to new treatments. My role is to develop and test new methods and conduct analyses. So an example of a particular method that I've developed is called partition, and it involves data reduction. So taking a large data set, reducing it to a smaller data set while still including most of the information. And this can be important in not only DNA variations, but other types of molecular measures, such as gene expression, which is a measure of, or it indicates essentially how much a gene is being translated into protein in a cell. Another type of data it might be useful for is epigenetics, which involve a molecular matrix around the DNA. And epigenetics also affects how genes are regulated in a cell. So another example of a method that I've developed is called the CIT, and it's essentially part of a class of methods called mediation analysis. And the focus and the objective of these methods is to distinguish correlation or simple correlation from cause-effect relationships. So, for instance, the CIT has gained some traction in studies that look at how DNA variation affects gene expression or epigenetics, and how those features in turn affect disease outcomes. Well, I first became interested in data analysis during my undergraduate work, at which time I was working part-time for doing research for the National Marine Fisheries Service in Alaska. And at the time I was taking a intro level statistics class, and it was a really great class. It was really interesting. And I was able to extend concepts I learned in the class to apply them to the research I was doing for the National Marine Fisheries Service. And that was in the early 90s, so soon after the Exxon Valdez oil spill, which occurred in Prince William Sound, Alaska. So I was able to not only do physical research in Prince William Sound, looking at the intertidal zone, collecting data from mussels that lived in the intertidal zone, but I was also able to do statistical analysis and extend what I learned to estimate growth rates in these mussels and how they were affected in areas that were oiled during the Exxon Valdez oil spill. And this work was really exciting, and it was eventually published. I really love research because there's those moments where you have the feeling of discovery. Those moments of discovery are the most exciting thing to me. So let's say I'm doing an analysis and identify a gene that affects response to treatment, or maybe it predicts survival, or I do research involving new statistical methods. So I test out these methods and I evaluate them in different ways. And occasionally I will discover that a new method is very successful in an area that other methods have failed, and that's really exciting. So yeah, it's these moments of discovery that make research fun.